and welcome to xcellogenbiotech. I'm Pavitra and today I'm here with the topic DIPA CRISPR. So before going on to DIPA CRISPR, we should discuss about CRISPR. So what is CRISPR? CRISPR is clustered regularly interspaced short palindromic repeats. So what is this clustered regularly interspaced short palindromic repeats? These are nothing but sequence of DNA which is derived from a bacteriophage. Who derives the bacteriophage DNA? It's a prokaryote. That is, a bacteriophage when infects a prokaryote, the prokaryote will derive the sequence of DNA from the bacteriophage. And later when the same kind of bacteriophage infects the prokaryote, what happens is this bacteriophage will be destroyed. That is, the bacteriophage DNA will be destroyed easily. So this is a kind of acquired immunity, isn't it? This technique has been used by biotechnology. This is a tool for biotechnology. For what? For, of course, genetic modification, gene editing. So, is a CRISPR alone used? No. There is another protein called Cas9, CRISPR associated protein. This protein along with CRISPR helps to identify and cleave a particular sequence of DNA. So CRISPR-Cas9, they both together act as a gene editing tool. So what does this gene editing tool do? It can introduce a sequence and also cut out a sequence. So isn't it a big tool? So now we are going to see how this CRISPR technique and DIPA technique was combined and used for insect gene editing. In a latest research, it was proven that DIPA CRISPR is an easy and simple way for insect gene editing. So let us move on to DIPA CRISPR. DIPA CRISPR or direct parental CRISPR. So now we know what is CRISPR, how it acts, how it is used and all. Now, what is direct parental CRISPR? So the conventional method of gene editing was done by microinjecting the gene of interest into the embryo of the organism. But not all the organism will preclude access to its embryo stage. That is, not every reproductive system preclude access to its embryo stage. To overcome this limitation, direct parental CRISPR was introduced. So here, microinjection is not done in the embryo. Instead of that, microinjection is done in the egg of the female adult. So in such organism, DIPA can be used. So this is the advantage of DIPA over conventional method. Cockroach female ovulates the oocyte into the gentilial atrium where it is fertilized. And later it is covered by a hard cover or case or ootheca, and it will remain as ootheca for many days or weeks. So here the Cas9 ribonucleoprotein was introduced into the main body cavity of the female adult cockroach. So this is how it was done. And DIPA CRISPR is one of the simplest method compared to the other conventional methods. It was recently proved. Using this method, there was a successful establishment of knockouts in cockroach. So why this method is simple? Because it takes less time. That is, it consumes less time, and we don't need specialist to do this method, a DIPA CRISPR? Because we don't want somebody who knows very clearly about the embryo stage. Here the micro injection is done directly into the egg of the female. So we don't need a person, a specialist who knows deeply about an embryo. Okay, that was about DIPA CRISPR. Thank you.